Okay, so hello everyone. Thanks Leelavati for the introduction and organizers for having me here. So I'll be talking about a histone modification and its role in interphase chromatin memory when the cell is dividing. So I'll not really spend much time on this because Sabri already introduced. But what I'd like to emphasize is that there is this hierarchical organization which is only true for the cells which are in interphase. And that is the period where cell spends most of its time because only like 5-10% of cells are there which are in mitosis. But interestingly, when mitosis is happening, the linear architecture is lost because you see the chromatin condensing, the hierarchical architecture is lost. What you see is that the chromatin is now linearly condensing such that the rod shaped structures are formed, and now you don't see any chromosome territories, domains or tags. So the question is, when this is happening, the transcription as well as the structure is lost during mitosis. How do the cell retain that? Because it's very important that in multicellular organism, the cell type specificity is maintained. So what happens is, if you consider a cell as a book and there are different pages which are genes, and you want to remember which gene has to express in that cell, what you would do is you would bookmark this. So this is called mitotic bookmarking, where you keep the transcription factor bound or certain histone modifications while most of them are being removed to maintain fast transcriptional activation in the next interphase. And this field is still growing, but we do have certain idea about how enhancers and promoters are retained in this context. The problem comes when you want to retain the structure. So if you look at this heat map from Decker's lab, so they did this high C in cells which were dividing. These are HeLa cells from, if you look at from early G1 to M phase. And then in a non-synchronous population, what you see here in these two plots is, you see beautiful AB domain alterations, which is lost in mitosis. You see again TADs, which are lost in mitosis. So the structure is not really captured with the high C that we are seeing here. So now we ask, is there a way to retain the chromatin structure as well as function? And we hypothesize that this can happen with the histone modification because they are quite abundant. So now acetylation and methylation of histones is being spoken about, where the retaining of these two. So acetylation to some extent, although it decreases, but methylation, which is consistent, people have spoken about how retaining those at enhancers and promoters can maintain some type of cell specificity. What is not really known is what happens in the context of histone phosphorylation. So we picked this modification H3S10p, and why we choose this is because it's quite abundant during the mitosis. As you see, it's the most conserved among the different phosphorylations. You see tetrahymena having H3S10p in the nuclei, the roundworms, Drosophila cells as well as mammalian cells. So all the cells that are dividing have in mitosis H3S10p being accumulating on the chromosomes. So what we ask is, if the increase in H3S10 phosphorylation that you are seeing during mitosis, it can help in maintaining the structural as well as the functional chromatin memory in all dividing cells. So for this we choose HeLa cells because there's a lot of data set available that we kept mining for this study. So HeLa cells, if you see here, these are the cells which are mostly in interphase. Only few cells are in mitosis. To enrich the mitotic cells, we treat them with nocodazole drug. And then we do a chip sequencing because we want to look at the enrichment of H3S10p on chromatin. So what you are seeing here is from asynchronous to mitotic condition, there is this fourfold enrichment in H3S10p on the chromatin. And the obvious question is, where is it present? So it turns out that it's mostly around TSS and at the promoters. If you compare from asynchronous to mitotic, you see increase in the promoter regions. And the same can be appreciated from this snapshot of browser, where you are seeing acetylation mark, which is H3K27 acetylation. Now this is an enhancer mark as well as a promoter mark. And if you compare these two peaks, this is around MYC gene, again a highly transcribing gene. So you see H3S10p increasing here, but not really any enrichment here. So, okay, in bulk we see more at promoters, but not in enhancers. But this was fine. What caught our attention was that once you start zooming out of the on the genome, you start noticing these domains of H3S10p. So H3S10p is forming these large size domains. and few are shown here by this arrow. So this is chromosome 11 from start to end, and you start seeing these domains. So next we asked, okay, do they overlap with any of the interphase structure? So interestingly, if you take high-C data from interphase cells in HeLa, A and B domain, what you're seeing here is that the H3S10p domains are overlapping with the active domain. Now remember, these domains in mitosis are completely removed, as I just mentioned in the introduction. So you are not seeing any A domain or tag in mitosis, but still you are able to have H3S10p on those domains. This is quantified here. So B domains don't really enrich much of H3S10p, but A domains do enrich. So what we are saying here is that in dividing cells, here it's HeLa, but we have checked it for another like different cell lines. So that in mitosis, the structure is not there, but the phosphorylation is increasing on the domains. But this was about the physical enrichment. What about the activity? So it turns out that if you divide these domains, now these domains are again from interphase, and we are dividing all the active domains based on H3S10p enrichment. So you are seeing high H3S10p, medium and low. So there's a grade of H3S10p in these domains. And if you go back and ask from the asynchronous population, so which means in interphase, if you take the H3K27 acetylation mark, which is again an active mark, you see the same grade of active mark deposition on these domains. So what it turns out is that H3S10p is not only physically marking these domains, but it is captivating the function as well. So along those lines, we also check the tags which are present in these domains, and it turns out if there's more activation, you will have stronger tags, stronger interactions, which is captured with this plot. So the higher the H3S10p, more the interactions and activation. Okay, this was good, but what is exactly H3S10p doing there? So there's literature that suggests that H3S10p might be involved in chromosome condensation or local compaction, depending on the context. So what we asked is, again, we took the high-C data from Decker's lab, who have done high-C in mitosis. And what we are doing here is, I would like you to just focus here. So we are taking all the tags which have high H10p, low H10p, or which is a random category, and we pile them up and then check the enrichment, intra-tag enrichment. So you can see here, the blue color indicates more intra-tag interactions and the red is the opposite. And the same is captured in this plot, which is distal to local interaction, again from the high-C data, where what we are seeing is that locally the tags which have higher H3S10p are more compacted, and this is the data from mitosis. So okay, I will just summarize this here. So in interphase, there is a structure, there is inactive domain, weak domain, and active domain. In mitosis, that structure is absent physically. H3S10p is deposited in a way that it recapitulates the activity of these domains when it's increasing in mitosis. And not just that, it is compacting the domains where it is more. So you are compacting the most active domain the most. Okay, but then the question was, what exactly is H3S10p doing? So if we deprive the cell of H3S10 phosphorylation in mitosis, what will happen? So what we do is, we make stable cell lines in HeLa with this construct. So we have HA tagged with HA tagged with H3 serine 10, and the serine 10 mutated to alanine or the aspartate. Now both of these mutants cannot get phosphorylated, but one is it doesn't carry any charge, and the other one has the charge. Okay, so the system is such that it's inducible. Only when we give doxycycline, the mutants or the variants are going to express. So what you're seeing here is, once I give doxycycline, the cells have the HA-H3, otherwise they won't have. And the beauty of this system is, once you have a set timeline of arrest and release, because we want to study that in the mitosis or the next interphase, how the absence of H3S10p will have any effect. So what we are doing is, only just before the mitosis that we want to capture the data, we are inducing these mutants. So each cell line will express these different mutants, and then we ask what is happening to the compaction in mitosis, and in the next interphase, how the function or the structure might be changing. So for this, the mutants, what they show is, if you compare the wild type with the A and D mutants, this is metaphase spread. So you're seeing the chromosomes are more elongated, so probably the compaction is now less, and because of which you see that there is a compaction defect. And it turns out this compaction defect is nucleosomal. And why I say that is, if we did H3 chip sequencing, which is standard in the field, so when you want to look at the mitotic compaction, you would do H3 chip seq, and you can appreciate here that although the differences are subtle, but you see that there is decrease in H3 incorporation in the domains that have high H3S10p. We check this for promoter as well, so which means that locally at the promoters as well as on a large scale at domains, even on the chromosomes, you are seeing a compaction defect in mitosis again. Now the question was, in the next interphase, what's happening? So for this we did ATAC-seq, where we are looking at the chromatin accessibility. Now the data that I haven't put here is, when you divide the genes based on their expression, you see H3S10p being highest on the highly active gene. And so we divided the genes based on their expression from low to high expression. And we are seeing that, again, this is subtle, but you see that there is increase in the accessibility in the next interphase. And this is the plot for the same. Interestingly, this is captured in the RNA-seq as well, because you are seeing that the genes which are highly expressing over the wild type, they are gaining some quotient of transcription. So what we are trying to say here is, H3S10p is important for the compaction of the domain locally as well as for the large scale A domains. Once it's absent, meaning the mutants of histones get incorporated, the H3 occupancy is probably, the H3 is unstable, because of which you see that the compaction does not happen anymore, which leads to, probably, it leads to more of the transcription machinery being retained there. As a result, there is an additional quotient of transcriptional activity, or maybe the factors or the modifications, which result in this hyperactivation of the already high transcribing genes. So with that, I would like to thank Sachin, who helped me with the experiment, the analysis and the conceptualization of the story. Arif, Rajat and Anurag were here. They helped me with few experiments, and all the Dimple lab members for their help. Special thanks to SIF and NGS at NCBS who make these experiments possible. Shrimonta and Avinchal from IISc who helped us with the MESC culture. Funding sources, and if you would like to read more, this is the QR code. I would be happy to take questions now.
So, so thank you very much for your interesting talk. Okay. So I'm wondering how much mutant, H3 mutant were expressed by induction? If you compare to the endogenous H3.
Right. So we have those plots, maybe I can show it later, but it is, let's say 1/5 or 1/6 of the endogenous one. So it's very less, but still we are seeing these defects.
Okay. Thank you.
Great stuff. I was curious why you don't see this phosphorylation on enhancers. If it is tracking the active chromatin, do you have any ideas on that?
Okay, I have like answers in two ways, not the exact answer. So this modification H3S10p, it gets deposited by mediation of HDAC recruitment on the chromatin. So it turns out more the acetylation, more the deacetylation with respect to mitosis, which will lead to more H3S10p deposition. So if you compare enhancers and promoters have it more. That's one guess that it might be more here. Other thing is, there are few papers which have suggested that cells bookmark the promoters more than the enhancers. So maybe it's just adding to that part.
So nice talk. So is there an RNA component in this? Because people have previously seen that, the RNA hybrids or R loops also accumulated a lot of H3S10p phosphorylation, and it was related with condensation of chromatin at the pericentromeric region. So my question is that if you treat your cells with RNase H or overexpress RNase H, will it overcome the problems you face by putting in the phosphorylation dead mutant? It's a complicated experiment, but just I'm asking if there's RNA component in your biology.
So, yes, the R-loop paper is there. We haven't really gone into that direction, but what we had tried is, if we were thinking more than any other thing, transcription, H3S10p deposition is more correlated to transcription. So we had tried these experiments where we will treat the cells with DRB, which will stop the transcription, and you start seeing less of H3S10p deposition. So there is an RNA angle, but exactly with respect to the condensation and in the mutants, if we can rescue, that would be a good experiment too. Right.
Does it work? So I was curious, so when you have your different mutants, what happens to the heterochromatin, and is it somehow also related to what's happened to the active part, or
yeah,
what's the crosstalk?
I was actually expecting this question because there's this phosphomethyl switch that people talk about. We surprisingly don't get much of H3S10p on B domains. There are some B domains which I see in my data that will have H3S10p, and they again are very strong. So maybe there is some association, but in the mutants or in the wild type, we don't really see heterochromatin changing or heterochromatin being marked by on a large scale. So maybe the time scale that we are picking, so we are picking the pro-metaphase cells. I think there is like maybe a dynamic factor to it, but yeah, that's there.
Okay,
Okay,
so
thank you so
let's
much.
thank
Yeah.
her again.